Today we're going to talk about genetics, evolution, development, and plasticity. So one big question in psychology is why are we the way that we are? Think of the stereotypical serial killer. Is this a person who is just born bad? Something about their genes and biology have turned them into a cold-blooded killer? Or was it their environment? Were certain things in the environment shaping them into the person that they eventually became? Or is it both, right? The answers to these questions are going to affect how we see laws and how we see society as a whole. Because if people can control the outcomes of their lives, right, then it is going to determine how much we can judge them for what they do. If we're just a product of our environments and our genes, things that we have no control over, then can we really be blamed for the things that we do? I don't know. We'll figure it out. Now, if you've dealt with a psychology class before, you know that the answer to any question that is it this or this, is always actually, it's both. So both our genes and environment interact to shape our behavior. The issue is it's going to depend on what specific thing that we're talking about. If you're talking about the color of your skin or the color of your hair, your genes have a pretty big impact on determining those things. Now, the environment can affect those things. If you've ever been in the sun too long and your hair started to change color as a result of that, your environment is shaping your genes. If you think about things like facial expressions, how we respond to specific emotional stimuli, right? Americans in general are a very smiley people, right? When we see strangers, we smile. So we seem much more friendly than sometimes we actually are. If you go to Russia and you smile at a stranger, they're going to look at you like you're a crazy person because our environment shapes how we behave. If you look at psychological disorders, then something like that is shaped by both your genes and your environment. If you have so for example, addiction, right? It's very heritable. So if addiction runs in your family, you're very likely to become an alcoholic, but your environment is going to shape that. If you are in an environment where there is never any alcohol and you go, 20, 30, 40 years of your life before ever trying a beer, more than likely you're not going to become an alcoholic. So there's this interaction between our genes and environment, and depending on what specific thing that we're looking at, we can see that our environment has a big effect on how we develop, or we can see that our genes play a primary role in how we develop. So, how we express emotions is very strongly rooted in genetics. Based off of our bone structure and musculature and all these things that are determined by our genes, that is going to determine how we express those emotions. So, the physical expression of the emotion is determined by our genes. So Gregor Mendel was a scientist who was studying pea plants, and he noticed that certain pea plants in combinations would transmit specific characteristics to the next generation. So this is where we got the theory of genes, something intrinsic to an organism that is transmitted generation to generation. So one thing that you should know is genes come in pairs. These little pairs are called alleles, and they are aligned along the chromosomes. If you remember chromosomes from biology, there are these little things that carry our genetic information. So these are the things that are being transmitted from generation to generation. So, when we're talking about our genes, we can't not talk about DNA. Everybody, you were expecting it, you were waiting for it, right? So, a gene is defined as a portion of a chromosome, and it's comprised of deoxyribonucleic acid. I was afraid that I was going to mess up on the pronunciation, but I killed it. Deoxyribonucleic acid, DNA. DNA serves as a model for the synthesis of ribonucleic acid. So it's a chemical that is the model for the synthesis of proteins. So what our genes are doing is we're just saying what is going to happen later, right? So it is a blueprint for the building of our body. So one strand of DNA might have some information that says, oh, you know what? We're going to make your hair blonde, or we're going to make it so that you're exceptionally tall or very short, right? A lot of who we are is encoded in our genes, encoded in our DNA, encoded in our RNA. So back in the day, you were just a bundle of cells. Oh, you were such a cute bundle of cells. A sperm met an ovum, and those two bits of genetic data combined with one another and started replicating. And that data was the blueprint for who you are today. So the proteins started forming specific structures of the body and differentiating, and they told other cells to do specific things. So uh, even though our DNA is so small and so tiny, it is the core of who we are and who we develop to be, who we develop into, who we develop to be, who we develop into, who we develop. The fact that I'm so obsessed with making sure that that the words that I say are like the exact right words is encoded in my DNA. Somebody else recording the same lecture wouldn't care. They just say something and that would be right. Based off of my DNA, based off of these proteins that were telling my brain to develop in a specific way, I'm much more likely to focus on small details and obsess about them. So here's a little bit about how DNA controls your development. So you have your DNA right here, and it replicates, right? And on some of the DNA, we have our RNA, right? And so it gives, based off of this code, which we'll talk about a little bit later, it's going to tell specific parts of the body to either become certain things, right? So maybe you have some parts of your RNA that are saying, okay, well, this is going to be the heart, right? So this is a heart. It is that it's exact. If you look inside your body, that's exactly how your heart operates, right? But maybe you also have something that tells your heart to develop a little weirdly than it should, right? And that's encoded in there. Then you also have enzymes that are controlling the chemical reactions that help your heart develop. So it's both telling the actual organ or parts of your body to develop, but also releasing chemicals that shape the development of the parts of your body. So, as you might know, you get genes from two parents, right? So, you might have an XX and an XY, and based on the combination of those genes, right, you might become male or female, but you're getting genes from both parents. So, uh, depending on the gene, you might get the same gene from both parents, meaning that you are getting a homozygous gene. So if you have a parent with very light skin and another parent with very light skin, that gene for your skin pigment is homozygous. But you could have a heterozygous gene, which means that you have two different genes for the same thing. So you might have a very light skin gene and a very dark skin gene. And depending on how your genes combine, that's going to determine how your body develops. So when it comes to genes, you have dominant genes, recessive genes, and intermediate genes. So a dominant gene, regardless of what gene it is paired with, is going to that gene is going to be what presents itself. So, for example, if there's a cat and there's a gene for really dark fur and there's a gene for really light fur, uh, if the dark fur gene is dominant, then regardless of where the other genetic material is coming from, you're going to have a cat with dark fur, right? Now, sometimes a gene can be recessive. So that light fur could be a recessive trait where you need two light furred parents in order to create a light furred kitty. Meow, meow, meow. Uh, there's also what's called an intermediate gene. So if you have a cat with like a black cat and you have a white cat, if those two cats have intermediate genes for fur color, then you're going to have a cat that's fur is a mixture of both. So you'll have a gray cat. So if black is dominant, then no matter what other thing is there, you're going to have a black cat. If black is recessive, then it's going to defer to what the other thing is. But if you have two recessive black parents, then you're going to have a black kitty no matter what. If it's intermediate, then the mixture of the two is going to determine the actual presentation. So here's an example of what we're talking about. Right here, you have a dominant gene. Here you have a recessive gene. Both parents have both a dominant and recessive gene. Depending on the combination, you could have a child who gets both dominant genes, and they have the sensitivity to taste a specific thing. You could have the child have both non-tasting genes, so they're not sensitive to a specific taste, or you can have either of these situations where they get one dominant and one recessive gene from the parent, and they are still able to taste the specific thing because the tasting gene is dominant. So obviously, it is not as simple as that. Just simply the combination of genes doesn't always determine the expression of genes. At some point, we'll talk about the difference between a genotype and a phenotype. The genotype is what is actually in the gene. The phenotype is the expression of the gene. So sometimes genes are only expressed a little bit. Sometimes they're expressed in some cells and not in others. Sometimes they only express themselves under certain circumstances. So this is a nice introduction into how genes work, but it's not everything. So we have two types of genes. We have autosomal genes, which are essentially all other genes that are not the sex-linked genes, and then we have our sex-linked genes, which are the genes that are located on the sex chromosomes. So for humans, we have two sex chromosomes. We have X and Y. And if you are biologically female, then you have XX. If you are biologically male, you have XY. There are other chromosomal combinations that happen in humans. And if you look at other species, some species don't have the same XXXY. They have Z's and other things. So sex chromosomal combinations that we're talking about right now are only applying to like 99% of humans, but not all of the species that exist in the world. I think some fungi have like 4,000 different genders. Once you get outside of like Homo sapiens, things get kooky. So during reproduction, women get a wonderful choice. They get to choose which gene they contribute. They can either contribute an X chromosome or an X chromosome, right? So when it comes to the ovum, there's very little determination of the gender of the child. Now, males, some sperm are going to be carrying an X chromosome, some will be carrying a Y chromosome. So the sperm essentially determines the sex of the child. So if you get an X from the sperm, you have an X and an X, then there's a genetic female. If you have an X and a Y, you have a genetic male. So the Y chromosome, just so you guys know, is smaller than the X chromosome. So if if you were to compare the two of them, you'd be like, "Oh, look at that cute little Y chromosome." So it's a very funny thing because men tend to think of themselves as very strong and powerful because of their Y chromosome, but it's just like a little guy, right? And you have this big old X chromosome that's like, "Hey, what are you doing over here, buddy?" So the Y chromosome has fewer proteins than the X chromosome. Uh, so when we're talking about sex-linked genes, usually we're looking at things that are on the X chromosome. So, for example, color blindness, specifically red-green color blindness. There are also what are called sex-limited genes, which are genes that are present in both sexes. So you could be a male and have something that only affects females, but the sex-limited gene only affects the you if you are the gender that is affected by that gene. So our genes replicate, and most of the time when they replicate, nothing weird happens. But sometimes our genes can change. So one way that they can change is due to a mutation. Either some radioactive isotope just like bumps something and changes something. Or sometimes there's an issue in the actual replication of the gene. So we have what are called microduplication and microdeletion, which is during that replication process. Sometimes a chromosome might appear more than it should or less than it should. So some researchers actually believe that schizophrenia is a result of certain brain relevant genes either being microduplicated, so they're appearing too much, or microdeleted, they're appearing too little. So there's a field called epigenetics, which is basically the idea that genes can express themselves in different ways. If you have two people with the exact same genes, you would expect them to be identical, like identical twins are, but not all identical twins are exactly the same. Now, some of this is because of the mutations and the replication issues that we've discussed before, but genes in general are not active all the time. So if you think about puberty, remember puberty? So fun, so easy. But there are genes that are responsible for puberty that were only active during that time in your life. They're not doing that anymore. There are certain genes that were super active when you were a child that allowed you to get rid of your baby teeth and grow at a pretty rapid pace that are not active now, right? So, depending on the time of day or the time of your life, certain genes are going to be active, other genes will not be. So, understanding how our genes express themselves is important to understanding who we are and how we develop. So our DNA kind of exists in these little wound up balls around these molecules called histones. Now, what can sometimes happen is different chemicals can react with the histones, which will loosen the DNA's grip around them, which will make more DNA available to be messed with by other chemicals. So let's say maybe there's some cocaine, so you got a little bit of that burger sugar just hanging out. What could happen is the DNA loosens its grip on the histones, which exposes more DNA to change. So things that occur in your body on a chemical level are going to make your DNA more susceptible to change. So whatever you do, whatever you experience, whether it's stress, so something psychological or certain chemicals, if you don't have a good nutritious diet, or if you are doing specific drugs, all of these things that go into your body can affect your genes, either in the short term or the long term. And the fun thing is these things in the environment that affect your genes are going to affect your environment in the future because your genes also shape your environment. So, one important thing to talk about when we discuss genes is heritability. How likely that gene's characteristics are going to pass on to the next generation. So, uh, there are lots of things, many behaviors that we monitor that are very heritable. Alcoholism, mental illnesses such as depression, schizophrenia, right? All these things can be very heritable. Now, sometimes the environment has a big effect on whether or not something is actually herited. So again, when we're talking about genes, we have to understand that the interaction between the genes and the environment determine how a specific gene presents itself. So one way that we can do this is looking at the difference between monozygotic twins, identical twins, so twins who have the exact same genes, and dizygotic twins, fraternal twins, twins who have the same parents but different sets of genes. If identical twins are exactly the same in all aspects, then we can say that genes have a strong impact on their environment, especially if they're completely different than dizygotic twins. If dizygotic twins and monozygotic twins are exactly the same, then it's more than likely the environment that has a strong impact on how they develop. You can also look at adopted children. Those are children whose genes don't match their environment. So if they are almost exactly like their adoptive parents, then the environment plays a much stronger role than their genes. One great thing about science is when we know that there's a gene that can be affected by the environment. If we change the environment, we can affect the gene for good. So there is a disorder referred to as PKU, which is an inability to metabolize a specific amino acid, phenylketonuria. And if you know that a child has a predisposition to PKU, you can put the child on a very strict and limited diet to prevent the negative side effects of PKU, which can include things like intellectual disability. So, a strong environmental intervention can affect our susceptibility to specific genes. So, one important thing to know is genes don't directly produce behaviors. Genes produce proteins that will increase the probability that specific behaviors will take place, right? So, something might make you more aggressive, more passive, or more whatever. So, they increase the probability of specific things, but they don't directly influence specific things. Sometimes they might go about doing something in an indirect way. So your gene might influence how you behave towards other people, which is going to make an effect on your environment, which is then going to affect you. So if your genes make people like you less, then you might, so maybe you're just generally have a tone that's very sarcastic, right? So whenever you talk to people, even though you're being genuine, people might assume that you're being sarcastic and they might treat you poorly. As a result, that gene is affecting your environment, and because people treat you poorly, it might affect your likelihood of developing depression. So your genes shape your behaviors indirectly, and then also shape the development of your genes further by shaping your environment. So, evolution is the change in certain characteristics of a species over time. Now, these changes can be positive or negative because when you talk about the idea of survival of the fittest, some changes occur that actually don't affect your fitness. So, if you think about the fact that some people might be born with an extra digit, right? So, most of us have 5 fingers, some of us might be born with a sixth one. Now, there's no evolutionary benefit for something like that. But because there's no detriment to having it, it continues on in our species. So there are all these changes that are happening, and they're small, little changes, but some of them stick around because there's no harm in having them. So evolution is relatively simple, right? Offspring resemble their parents in very clear and observable ways, but because we know that genes can change very slightly, from generation to generation, you might see very slight variations. Over time, those variations are going to seem very large and drastic, which is a reason why fish and zebras are so different, right? But in the short term, you're seeing very slight differences. So some individuals are more likely to reproduce than others. So depending on what things are determined as desirable or what things allow a certain organism to get more mates, right? So, for example, it could just be something as simple as fertility. If everyone has the same amount of sex, but some people are more fertile than others, those types of organisms are going to have more offspring. And then that fertility is going to be passed on to further offspring, making that specific trait more common. Now, you've probably heard of natural selection, which is just the idea that in nature, things will figure themselves out and the fittest, heavy air quotes, will continue on. But there's also the idea of artificial selection. So if you look at the difference between the corn that you usually buy at the grocery store versus wild corn, right? It's completely different because we've specifically bred corn to get the most desirable traits. Dog breeds are the same thing. We have chosen the quote unquote best genes that we want to carry on, and we only allow those specific breeds to, or those specific like dogs to breed with each other so we can get the best characteristics. So when you hear the term genetically modified organism, a lot of people think of like a scientist in a lab, like playing with DNA using some scientific tool. But we've been genetically modifying organisms for the entire length of agriculture. We've been selecting the best crop or the best animals and breeding them with each other in order to get future generations of better stock. So that's artificial selection. That's man-made intervention. So there are some misconceptions about evolution, which are one is the theory of Lamarckian evolution, which is the idea that certain changes in the body that don't actually affect the genes can be passed on to a child. So, for example, let's say I marry somebody, and both me and my spouse have our appendices removed before we give birth to a child, and by we I mean she, before a child is conceived by us. So the in Lamarckian evolution, the idea is that lack of an appendix would be transferred to our child, which is not how evolution works. That's a surgical intervention that doesn't actually involve genes. Now, the likelihood of our child having a appendicitis is probably more likely than another child because that child had two parents with appendicitis at some point. But that child isn't going to be born without an appendix just because its parents didn't have one. Also, there's this idea that humans have stopped evolving, but we're changing constantly. There are some humans that have developed a genetic resistance to HIV. That's a product of evolution. That's a product of these very slight mutations that are happening constantly. Evolution also doesn't always mean improvement. There are some evolutions that are not great. So, for example, flat-footedness, right? That is a human evolution. Some people, myself included, so my flat-footed people out there in the house, give me a yay yay. But that is a form of really terrible human evolution because it makes it harder to walk and run and makes it more likely that your knees are going to be damaged over time. It's not the best form of evolution. And not all evolution is for the benefit of the individual or the species. So, those are a few misconceptions about evolution, but now you know that evolution can be for good or for bad. So one explanation for the reason why we are so different from a lot of the other species that are around, why we think ourselves so superior, is because of our superior brains. If you look at the structure of our brains in comparison to other animal brains, we have a lot more mass, especially in the cortical areas. So there were certain things that we started doing that provided our brain with the nutrition that it needed to develop. We started cooking food, which made food easier to digest. Because we hunted in coordinated groups, we were more efficient and were able to get more food. And also, we probably just developed better glucose transport systems, and as you know, glucose is the power supply for the brain. So if the brain is getting more power, it can do more. So now we transition into what is called evolutionary psychology, which is the idea that certain things in our evolution made us more adaptable to the environment. So, for example, the fact that we developed this deep capacity for social interaction led us to develop tribes and societies. That interdependence on one another allowed us to form the formation of things that would allow that the our foundational structures of society would be built upon. So if people specialize, right? If some people hunt, if some people stay home, if some people build things, if some people use those things that are built to do other things, right? That social aspect of our evolution is key to how we developed. So all these specific things that are biological and social that are key to our evolution are the reason why we developed the way that we did. If a lot of those things were different, then we wouldn't have developed the same way. If we didn't respond to language in the same way, if we didn't have these very developed prefrontal cortexes that, or sorry, cortices that allowed us to plan and make proper judgments, we wouldn't have the society that we have today. So, the difficult thing about evolution versus societal influences is sometimes it's very hard to know what is key to our biology, right? And what is just based off of society. Because society, after a certain point, developed past our evolution. So, things like the length of our lifespan, or how we view sexual promiscuity, or even altruistic behavior, doing things ideally in a selfless manner. Do these have roots in our evolution, or are they developed by society, and do we inherit them not because they're encoded in our genes, but because we've been taught them? It's hard to know. So a gene can only spread if it is allowed to spread. So if that person or organism is spreading its gene more so than other genes, then that gene is going to become more dominant. Now, there's one theory that altruistic groups who cooperate work better together, and that allows specific genes to work. But it kind of falls apart because the idea of group selection depends on everyone in a group cooperating, and most animals don't cooperate to the same extent that humans do. But more than likely, what we see is a lot of kin selection, which is the idea that it's not just based off of who's the nicest and everybody getting along, but organisms are going to select other organisms to mate with based off of the genes that they view as more beneficial, right? So if you think of the male peacock that shows its beautiful plumage, a female peacock goes, yeah, I want that, right? Peacocks with lots of plumage and beautiful coloring are the ones that are going to spread their genes more because of kin selection. So there's the idea of reciprocal altruism, which is the idea that if you help somebody, they will help you, right? It only works if you trust that the other person is going to do that. So if I, on the first exam, give a student a Scantron, and I'm doing it because I believe that at some point someone else will give me a Scantron if I need it, then it's based off of the belief that somebody will take care of me when I need it. If you think about like social programs, right? Some people go, well, I don't want, to pay for other people's health care because that's money out of my pocket. But the idea is if we all pay for everybody's everything, right? And not everything, but if we all contribute to healthcare and bring the cost of it down for everyone, then everyone has it when they need it, right? But the worry is that when you need it, right, it won't be the quality that you're expecting, or you'll never need it and you'll be paying for other people's stuff without getting anything in return. So the development of the brain depends on two things: maturation, how it develops over time and how it differentiates and grows, and learning, the acquisition of new information and the association of that information with existing information. So we understand this by looking at how our neurons develop, how they connect with one another, and how new experiences affect that development. So if we're looking at our brain, right, this is your brain, these are your little eyes, which haven't developed yet, right? The dorsal surface starts to thicken, and there's a little tube that forms under that, because the dorsal surface has thickened. It is filled with fluids, and this dorsal portion enlarges and becomes your hindbrain, your midbrain, and your forebrain, and this little tube becomes your spinal cord. So this is what you looked like once, right? So as we can see over time, things start to differentiate, and now we have what's going to become the spine, and we have a little heart developing here, and here's your little brain. And soon, one day, it becomes a big brain. I'm very good at drawing brains, as you can tell. And now we can see more of the differentiation, right? So this is what we expect the brain to look like, but it has to start somewhere. So we have this long tube, right? And then the forebrain starts to grow, and it will grow over the other parts of the brain, right? You can see now in comparison, it's much larger than the midbrain and the hindbrain. And once it fully differentiates itself, we see that at birth, you have the brain that you're going to have as far as how the anatomy looks. And the little midbrain gets hidden under here, but it's still there. And these cortical structures have begun to, like all the folds that we're used to in the brain are there. So, as mentioned before, in the center, right, as everything's forming around it, we have this fluid-filled area. The fluid is our cerebral spinal fluid, and that differentiates itself into our spine and our spinal cord and the four ventricles of the brain. So, our brain develops very rapidly. At birth, it weighs about 350 grams, and by the time you get to your first birthday, it almost triples in size. And that shows how rapidly the brain is developing. In comparison, once you are an adult, it's not doubling or tripling, it just increases very slightly in size as certain connections are reinforced and other needless connections are trimmed down. So, the development of neurons in the brain involves the following processes: proliferation, migration, differentiation, myelination, and synaptogenesis. So, proliferation is the creation of new cells and new neurons in the brain. This primarily occurs very early on in life. So the cells lining those ventricles, which are formed outside of that tube that becomes the ventricles and the spinal cord, uh, they start to divide. Some of them become stem cells that continue to divide. Others become neurons or glia that move to other locations. So the movement of the glia and neurons to their eventual locations is called migration. Some don't reach their destinations until adulthood, so this isn't a quick process. Sometimes this can be a very slow process for some of these cells. They occur in a variety of directions, and it is because there are chemicals called immunoglobulins and chemokines that guide that migration. So once they've migrated, they start to differentiate themselves. So they start to become the neurons or glial cells that they are supposed to be. And that is differentiation. Then we have myelination, which is the process through which glia produce the myelin that aids in the transmission of neural impulses. We've talked about saltatory conduction at the nodes of Ranvier, right? So the signal can just jump from node to node, which is much faster and much stronger than traveling down the axon. So the myelination first occurs in the spinal cord and then goes up the hindbrain to the midbrain and the forebrain. And it occurs over decades, right? If you remember, the prefrontal cortex is the last part of the brain to develop. So if development is happening starting from the spinal cord and moving forward, this myelination is going to happen here last. And then we have synaptogenesis, and that is the formation of these connections between our different neurons, right? So neurons are constantly creating connections, and certain connections, if they haven't been used in a while, they will fade away. And they're actually little cells that will come by and clean up any mess that is left over. But the especially in early development, you're forming lots and lots of connections. The older you get, the fewer connections will be made. That's why it's significantly easier to learn something like a new language when you are younger, because your brain is more plastic, it's more ready to adjust to new information, form new connections. So it was originally believed that after your early development, no new neurons were formed, but that's actually not true. So one thing that we have in our brain are stem cells, which are undifferentiated cells that still stick around in our brain and are able to later in life differentiate themselves into glia cells or neurons if necessary. Also, your olfactory receptors, responsible for smell, will continually replace dying ones because after a while they wear themselves out and they need to be replaced. Part of the reason why we're able to continue to learn as adults is because the stem cells in our hippocampus, which is responsible for the formation of new memories, will differentiate and help form new connections to help consolidate new learned material. A few things to remember: different cells have different lifespans. So, our skin cells are the newest, they last for about a year. Heart cells tend to be as old as you. When we're looking at the cells in our cerebral cortices, those don't form many new neurons after birth. So when axons are trying to make connections, there are all these chemicals present in the brain, and basically the axon will reach out and be attracted to certain chemicals, but be repulsed by different chemicals, right? So they find their way by following this path of chemicals. So here's a little newt, and these are its little newt eyes, and we've got some little neuronal pathways going towards the optic tectum. So up here, we have what the connections normally look like between the retina and the optic tectum, where all the ocular data is being received. Now, what Sperry did was he cut the connection, right? And then he rotated the eye. Now, what you would expect is for the connections to regrow and be flipped, right? So if it was A, B, C, and he flipped it, then they would connect B, C, A. But when he flipped them, the connections actually went back to where they should have been before. So even though A was lower, right, it still went high to where it actually was, which shows that these neuronal pathways, these axons, know where to go, and it's based off of the chemical trail that tells them to go dorsal or posterior or anterior or ventral. So once again, the axons know which path to follow based off of specific chemical gradients. So axons will reach a neuron, and what happens is, the action potential goes down, but the postsynaptic cells will strengthen the connection with some cells and eliminate the connection with others. So that's how these connections are reinforced. Based off of what signals are being sent, that's going to determine what things are reinforced and what things are being cut off. If something is coming from this sending neuron, and it continues to create action potentials, this pathway is going to be reinforced. If this one, is like that like one person who's like interested in you and like text you all the time, and then they like ghost you for 5 months, you're going to delete that number. Some people refer to this as neural Darwinism, the survival of the fittest connections. So the brain is going to form more connections than it actually needs. But the things that are most necessary, the things that are used the most, are going to survive. So if you think about certain like specifics about language, right? So for example, in Spanish, you have the ere and the ere, right? Right there. So if I speak Spanish and I have an ere pathway, then, and there's also my ere pathway, then there's going to be somebody who's like, your ere sound like eres, right? And I'm going to be like, sorry, I haven't been practicing. So those neuronal pathways are being cut off. So the things that are used most often are going to stay around. Now, if you have lived in a community where you've never had to roll your R, at some point in your development, you probably would have been able to make a really great R, right? R, oh, the more I do it, the worse it gets. But because you never used it, that pathway dies. But other things, like the difference between like mayo and aioli, right? I don't know. Aioli, how's it spelled? I don't know how to spell aioli, but I do know how to erre, right? So depending on what is used, right, that's going to determine what is reinforced. By the way, it's aioli, Aioli. That is way too many vowels in a word. I'm just going to say that now. Aioli is canceled. So when we're looking at this calling of nerves, because we overproduce, we should talk about the nerve growth factor, which is a protein that is released by muscles that promotes the survival and growth of specific axons. So we have maybe like four different neurons that their axons are going to this muscle, but there are only two places where they need to connect. So out of these four, the NGF goes, I'm going to give you a rose, and I'm going to give you a rose. You have to go home. So the communication between these two is what determines how long or which neurons survive. Then we also have what are called neurotropins, which promote the survival and activity of neurons. So if you are not exposed to neurotropins, then you're probably going to experience cell death. So a in a healthy nervous system, you're going to have released enough neurotropins to tell the neurons that you want to survive that you like them, right? So all of these neurons get a gold star, A plus, right? These are the best neurons, so they are going to survive. All these other ones over here, we don't care about them. So neuronal death is normal. It is a normal part of our brain's healthy development. We overproduce, we make connections, and the things that work stay around, the things that are not needed die. Fun fact, the visual cortex is actually thicker in blind people because there's no visual stimuli. So there's no balance between the things that are being used and the things that are not being used. So the brain has a much harder time pruning out ineffective neurons because nothing is being stimulated. So there's no balance, there's no difference between the overstimulated and the understimulated. So you have heard this probably from some drug campaign somewhere, but your brain is vulnerable. You only got one, so you got to take care of it, right? Especially in early stages of brain development, prenatal development, and infant development, it's very important to make sure that your brain is getting the right nutrients and not being exposed to harmful chemicals. Even an issue in one gene can lead to severe birth defects. If it's the gene that tells your occipital lobe to develop properly, right, then you might experience issues with visual processing. Or if your temporal lobe isn't processing correctly, then you might have issues with hearing or auditory processing. So any chemical distortions in the brain early in development can cause significant impairment and developmental issues. So as I'm recording this, I am sipping ever so slightly from a glass of whiskey. So I was about to reach for this glass, and then I went, no, I'm going to take a break from drinking for just a few slides because alcohol can have very detrimental effects on the brain. One of these effects, which you might have heard of before, is called fetal alcohol syndrome. So if a mother is drinking heavily during pregnancy, heavily being the key word. So one glass of wine once during the 9.5-ish months of fetal development, probably not going to do much harm. Drinking a bottle of vodka a day and washing it down with another bottle of vodka, not going to be great for the development of the child. So some of the symptoms of fetal alcohol syndrome: hyperactivity and impulsiveness, difficulty maintaining attention, varying degrees of borderline intellectual deficiency, motor problems and heart defects, and facial abnormalities. So if you remember our little neurons, right? They have dendrites. In fetal alcohol syndrome, these dendrites are much shorter and they have fewer branches. Let me draw one that has fewer branches. There we go. That's good enough. So the exposure to alcohol in the fetus suppresses glutamate and enhances the release of GABA. If you remember, GABA is inhibitory. So it's telling the neurons to not make, to it's slowing down action potentials, which means that the brain isn't making the same connections because if the receiving neurons know to strengthen a connection based off of the number of signals that they get. If they're not receiving as many signals, they're going to cull some of those connections. So less excitation as a result of alcohol is going to mean that there's slower development in the brain. That's why alcohol is so dangerous to the developing brain. So, as we have mentioned before, the neurons in the brain differentiate and serve different purposes. If you were to take a neuron before it starts to differentiate itself and you put it in a different part of the brain, it will develop the properties of that new location. If it's transplanted in the middle of that differentiation process, then it will retain some of its old properties, but still develop some new properties of that new location. So what's happening with this ferret is that it's been trained that when it sees a light on its left, which stimulates the right hemisphere, it should turn to the right. Now, when its brain is rewired and it sees the light on its right side, it should turn to the left. But because the brain has been rewired and switched, it turns to the right. So neurons, even when placed in a new area, still retain some of the properties that they had before. So our brain is continually reorganizing itself. And so even though we're not creating new neurons, new connections, structures, and other modifications are happening through the lifetime. So the spines on our dendrites are continually growing, and any changes to those spines is symbolic of a new connection. And that relates to learning. So here we can see some changes in the dendritic trees of two different mouse neurons, right? So you can see some extra spines here. We can see some big changes from here to here, right? So especially in this one, there are lots more branches on this left side. So the neurons continue to change over time even in just the span of a month. So, a stimulating environment is going to foster more neuronal growth. So, if you have a developing brain like a child and you talk and read and sing to it, like the commercials say, then that's going to foster more brain development. A stimulating environment for a child is so important to development. When you see children that are neglected, which is honestly the worst thing that you can do for the development of a child, uh, they don't get enough stimulation. So these new neural pathways and these branches and spikes cannot form. So stimulating environment, physical activity, all these things that we say are helpful to us, right? We know that physical activity is good for our bodies, but it's also good for our brains. So an enriching environment, even though neurons will die, new ones will come to take their place, and you'll see that improvement in learning and memory. So there was once this idea that if you teach a child a difficult concept, that will enhance their intelligence in other areas. So if I'm teaching a child calculus, they're going to have a much easier time with spelling. That's not actually how the brain works. But if you are teaching somebody one skill, their skill in other related areas will improve. So if I teach a child calculus and later have them do accounting, which is math based, they'll have a much easier time with that. So, if you've ever heard the idea that if you lose one sense, the other senses get stronger, there is genetic basis for that, because what ends up happening is the cells that aren't being used for one specific task differentiate themselves and aid with other tasks. So in the case of blind people whose occipital lobes are not responding to visual information, touch information will be processed through the occipital cortex. So people who are blind from birth are going to have a much easier time discriminating between objects by touch than those who rely primarily on vision to determine the differences between objects. So, especially, or if you look at the brain using transcranial magnetic stimulation or TMS, you'll see that inactive brain areas that shouldn't be active because they're not responding to sight are being used to discriminate between tactile stimuli. So those brain areas are being used, but they're just being used for something that they normally aren't used for. So you can see these changes and specialization in the brain in other situations. So for example, the temporal lobe, which is responsible for hearing and auditory processing, is 30% larger in professional musicians than in non-musicians. And these changes can even be seen when you scan the brain. So the things that are responsible for motor coordination, right? The aspects that are responsible for vision, so for example, reading, music, all these aspects of the brain that are responsible for being a musician, you're going to see much larger, more developed areas in people who practice music versus those who don't. And even though we know that adult experiences can modify brain anatomy, we're still not completely sure if it's significant enough to be observed with an MRI or similar technology. So in some cases, you can overpractice and form too many connections. So focal hand dystonia, or musician's cramp, sometimes also referred to as writer's cramp, is a condition where your brain has reorganized too much. So if you're practicing too extensively, so let's say you're a guitarist, your fingers might be clumsy, you might get tired more easily, or they might make involuntary movements. So if you have not seen this before, this is what is called the homunculus, and it shows how different parts of the somatosensory cortex are related to different muscle groups. So here are the hands and fingers, right? And in a normal brain, you have a good chunk of your brain that's responsible for the coordination of your hands and finger movements because we use them for so much. And then in the musician's brain or a writer's brain or anybody who's using their hands more frequently than the normal person, you're seeing a much wider spread because more of the brain is being used for a specific activity. So I've mentioned this before, but our prefrontal cortex, right here, is related to our ability to make judgments, to plan, impulse control. So because adolescents don't have a fully developed prefrontal cortex, they tend to be more impulsive than adults. So when it comes to drinking, risky driving, sex, all those fun things, they might be more impulsive. They might not fully think things through. They also don't really care as much about the future. So it's much harder to tell a adolescent, hey, sit up straight. When you're 30, you're going to have back problems because they don't have that same ability to plan as somebody who is maybe 27 and going, oh, I really should take care of myself, right? So that's a result of brain development. So one thing to be cautious of is making sweeping generalizations. So it's not that adolescents can never plan or make decisions or think about the future. You can probably think of many adolescents who are very concerned with the future, but we're just looking in comparison, right? Also, while the prefrontal cortex is primary in those types of planning and decision making behaviors, it's not just solely focused on the prefrontal cortex. So it's not the full explanation for why adolescents tend to be more impulsive. On the flip side, old age. So over time, neurons lose their synapses, and the remaining synapses will change more slowly, and sometimes they won't respond as well to stimulation. Brain structures begin to lose volume. And also, as we've talked about before, over time, new connections aren't forming as quickly. So the loss of brain mass and connections and structures, coupled with the slowness of renewal, is why you experience cognitive slowness. So not necessarily Alzheimer's or dementia, but just the difficulty in learning new things or adapting to new situations. So even though our brain is very sensitive, it is still plastic. So even after brain damage, we can recover. Some of the recovery is due to the cells that we have in our brain being available to generate new cells. So if we have access to stem cells, new neurons and new connections can be formed. But also sometimes what happens is some parts of the brain will compensate for the damaged parts of the brain. So when we're looking at what happens in the damaged brain during recovery, it looks very similar to what the normal brain is doing, forming new connections, pruning connections that aren't working, trying new things, right? So the younger your brain is, the more plastic it's going to be, the easier it's going to be to bounce back. But still, in later years, your brain has some ability to be flexible. So there are lots of things that can happen to your brain: tumors, infections, exposure to toxic substances or radiation, degenerative diseases like Alzheimer's, closed head injuries, so concussions, right? Getting into a car accident. All things like this are going to affect your brain and its ability to do its normal functions. So a closed-headed injury is one that is a blow to the head that does not puncture the brain. So what is often happening, one thing that we don't remember about our skull is that it's very hard and our brain is very soft. So even though our skull protects our brain from very severe damage, right? The fact that our brain is just basically jello in a hard shell, any blow to the head is going to cause the brain to slam against the skull, which is hard. So you can still experience damage to the cortices or other parts of the brain, which would be less or it would be more damaging if the skull weren't there, but it's still damage nonetheless. So this is one of the most common sources of brain injury in young adults: car accidents, concussions from sports, things like that. And in severe cases, as you might imagine, recovery can be slow, and in some cases, people never return to their full level of functioning. In the elderly population, more than likely what you're seeing are strokes or any sort of cerebral vascular accident. So any issue with blood flow to the brain, which, as you might imagine, the brain likes blood flow. So if these are real fun. I love talking about brain damage because it always makes me feel like really scared. It makes me want to like hug my little brain and say, "I will protect you forever, little guy." But over here we have somebody who experienced a stroke and died immediately, and you can see that there's swelling compared to this side. We have somebody who over here who experienced stroke and continued to live on, and you can see the reduced mass from after the stroke. And you want to guess what happened here? All this damage that went straight through the brain. That is what a gunshot does to your brain. I always tell you guys, don't do drugs, avoid gunshots. There are two types of strokes: ischemic strokes or ischemia, which is when one of the blood pathways to the brain is blocked. So if blood can't get to the brain, you'll have an ischemic stroke, and your brain can't get oxygen or glucose. And fun fact, in case you did not know, your brain does need oxygen and glucose. That will be on the test. And if it's not on the test, just know it for life. There are also hemorrhages. Hemorrhages, hemorrhages, hemorrhages is because it's one hemorrhage, and if you have multiple hemorrhages, man, I don't think that word is impossible to say. Don't try it, and don't you dare record it and then email me a recording of you saying it, because that would be rude. It's I'm just gonna say and call it for humanity. It's impossible to say. But a hemorrhage is not when it's blocked, but when the artery ruptures and you get bleeding everywhere, and that is not fun. So the neurons are flooded with too much blood and calcium and oxygen and other chemicals, which is equally bad. So remember, our bodies work in balance. You want enough oxygen and stuff, but if you have too much, oh boy, that's a bad. So, strokes can also cause edema, which is too much fluid in the brain. And if the brain swells, if you remember, your brain is in your skull, and your skull isn't very flexible. So, if your brain is pushing against the skull, there's nowhere for it to go. So, it's going to damage itself. This could also lead to disruption of your sodium potassium pump, leading to too much potassium inside of your neurons, disrupting the action potential process, throwing off that chemical and electrical charged balance, which isn't great. So, really, your brain is very sensitive. So again, too much potassium, right? we have a balance of potassium for a reason. Too much is going to trigger more glutamate to be released, which means that your neurons are going to be overstimulated, which you don't want anything to be overstimulated. So too much potassium is going to cause issues, and it could end up killing neurons. So it's a long way around, but even though you might be like, oh, a little potassium in the cell isn't terrible, no. That, in addition to the damage to the cells caused from excess blood or not enough oxygen, could also just cause the cells to die. So we have to be careful. So don't go damaging your brain, okay, kids? So if you have a stroke, don't worry. You can use a drug called tissue plasminogenic activator, which helps break up blood clots and can reduce the effects of a stroke. Two things that it does. One, it blocks your glutamate synapses, so it prevents your neurons from overfiring, and it prevents calcium entry. You don't want too much calcium in the cell, so by blocking glutamate and calcium, it reduces the damage from the stroke. You can also cool the brain. By cooling the brain, you're slowing down those normal metabolic processes. You're also slowing down the overstimulation and the apoptosis, which is preventing the brain from trimming those connections and trying to kill itself. Also, don't tell anybody that I told you, but cannabinoids have been shown to potentially minimize cell loss after a brain stroke. So don't tell them Professor sent you, but if you want to save your brain, you got to smoke a little ganja. You know what I'm saying? Uh, I wish I had the rights to some reggae music that would just be playing in the background for the rest of the video, but I can't do that. But the reason for that is because cannabinoids, like THC, are anti-inflammatory. So just so you know about the research, they are most effective when you take them before the stroke. So if you think that you're about to have a stroke, toke up, my friends. And that's science. That's not me telling you to do drugs. This is me telling you to save your brain. So one of the rough things about brain damage is if you have multiple cells that are in a chain, one, two, three, and two is damaged, even though one and three are working, because of the damage to two, this chain is disrupted. So the after a stroke or any type of brain damage, you might see what's called diastasis, 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 dia. It's called diastasis. Nope, that's not a word. That's not a real word. But that is the decreased activity of surviving neurons after damage to other neurons. So that two was destroyed, right? So one and three can't do what they were doing before. Diacheses, diacheses. I think it's diacheses, diacheses. It's when you have like Gruyere and also blue cheese, die cheeses. Uh, I, I hope somebody's gonna listen to this and be like, I don't think this guy knows anything about how the brain works. Correct. So that's sarcasm. I'm not sure. It's hard if usually my face indicates that I'm being sarcastic, so you just have to get it just off of the tone of my voice. So hopefully you haven't experienced some two cheeses and you can't figure out that I'm being super sarcastic right now. So anyway, back to the brain, which mine is working so good right now. Drugs, specifically stimulants, can help stimulate activity in healthy regions of a brain after a stroke. This makes sense. If the issue is that your brain is underperforming. A drug that tells your brain to do a little bit more, maybe stimulates a little bit of release of glutamate, right? That's going to help your brain operate a little bit better. In case you actually want to know how that word is pronounced, it's diascasis, okay? Diascasis. Hope you're happy now. So cell bodies cannot be replaced. Axons can grow back under certain circumstances. So if you had damage, like we talked about before, sometimes what can happen is the scar tissue, where the damage occurs, prevents new connections from forming because the axons can't get over the gap. Also, what can happen is the neurons will just pull apart from one another. Sorry, arrow should go in the opposite direction because they're like, well, we give up. There's no point in trying, so they just don't try. The glial cells that are around could also react to the damage and inhibit the growth of axons. So this little glial cell over here might go, "Hey guys, let's just not try this. Let me just release some chemicals and tell you that it's not worth it." Right now, we've been doing some research into building little protein bridges, which would help the axons connect to one another, but that is still a bit of a ways off. So the fun thing about these axons that are growing, right? They will still try to make connections, which is great. We want them to do that. Unfortunately, what can happen is they make a connection that doesn't really do anything exciting or does something that's really hurtful, right? We want them to reconnect to the thing that we had before, which was super cool and great. But unfortunately, that is what happens. Now, over time, uh, these sometimes these will repair themselves, but you can have connections that don't serve any positive purpose that create this positive feedback loop where they continue to reinforce themselves by activating themselves. So you have this very problematic connection that you cannot trim. Brains are rough. Like they're just not, like they're very helpful, but sometimes, sometimes So here we have two different axons, and this one unfortunately gets injured. So this one compensates by sprouting out and filling those vacancies. Kind of like a stepdad who comes in and takes care of this little mama neuron while papa neurons out. I think that's how stepdads work. They just come in for like a week or two. Is that how stepdads work? I don't know. So if the receptors are used to getting lots of neurotransmitter action, uh, what can happen is if a couple of these break down and you're only getting one axon that's sending neurotransmitters, these receptors might become super sensitive. And that sensitivity is because they're still trying to have their action potentials, but they're kind of, lowering their expectations. They're like, well, usually I want my neurotransmitters to be 6'3", but I'm fine if my neurotransmitters are 5'8", right? So that increased sensitivity is compensating for the decreased input. But sometimes what happens is they become too sensitive, and that means that they are now susceptible to almost any stimulation, right? And that can cause issues like chronic pain. So Real quick lesson from neurons. When you drop your expectations and you just let anything affect you, it's going to cause you some chronic pain. You thought that you're going to learn about biopsychology? I taught you some rules about dating. Respect yourself. So, if you've ever heard of phantom limb syndrome, sometimes an amputee will still continue to feel sensations in the area that has been amputated. So, if you've lost your hand or arm, you might still feel an itch or sensation, even though there's no actual sensory input for that arm or hand. And the reason for that is because of the reorganized nerves. So other parts of your brain and other sensory input is stimulating those old pathways. So, in the case of somebody with a phantom limb, you feel a cold breeze, and it might trigger an itch on your elbow. Interestingly enough, the use of an artificial limb can reduce the likelihood of experiencing phantom limb. Because, for example, sometimes things like an itch, right? Even if you're not actually itching the affected area, the action of itching soothes the sensation because of how our brains are trained. Sometimes just thinking about doing it, right, can even if you're not providing that actual physical sensation, or if you're doing something similar, it can be just as soothing. Our brains are these wonderfully complicated things that in extreme situations can do these very bizarre actions that can also be treated through unconventional ways. And that's part of the reason why our brains are so cool. Just going to give a quick shout out to brains real quick. Give it up for your brains. There we go. Clap it up. Clap it up for brains. Yes. Yes, brains. Yes. If you're not clapping with me, you're a jerk. So here we can see the hand, right, mapped on the somatosensory cortex, but after the amputation of this digit right here, D3. That's supposed to be a D. I don't look like a P. Just trust me and believe that it's a D, right? The D4 and D2 areas grow in order to compensate for that missing input. And look, ooh, spooky. It disappeared like a ghost. Ooh. And then here we have, so if this arm were amputated, somebody getting a touch on the cheek might stimulate these same pathways and cause a phantom sensation. So sometimes a limb might lose its afferentiated neurons, which are the ones going. This is somebody's arm. It's a very good arm. The ones that are going back into the brain. So what happens is there's not as much feedback. So usually you're sending to the muscle, right? Your efferent neurons, and then you're receiving input back. And what happens is they are less effective because they don't have all the information. Now, what can happen is people just learn to compensate. You can either compensate by using the other arm or use that arm or limb in a different way. So scientists just like to cut things in animals and see what happens. So if you were to cut the dorsal root of a nerve pathway, then what would happen is you'd still have the ventral root, which allows you to control the muscle, but you wouldn't have the sensory input. So I might be able to move my hand to catch a ball, which is ventral, right? But the actual feeling of the ball landing in my hand and the impact of that, I might not be able to adequately respond to. So all this to say is our brains are endlessly complicated. If you're looking at the cells, if you're looking at the cortices as a whole, if you're looking at the brain as a whole, if you're looking at our peripheral nervous system, all of these things work in concert to make our lives do what we do, make us do what we do. So, damage to any part of this, usually there's some level of plasticity and ability to recover. But sometimes, unfortunately, depending on the type of damage, there isn't. So that's that's it. We did it. It's done. Woo! I'm going to take a break. I'm going to grab that glass of whiskey that I put down when we were talking about fetal alcohol syndrome. Oh no, now I'm thinking about it again, and now I don't want it. Okay, take care, guys.